Welcome to my previous year question discussion 2023. The next question, what is fixative and mention its one use. First of all, we should know what is fixation. So, fixation, it is the first step in sample preparation and has the aim of preserving structure of living tissues in its original state. Okay, fixation makes cells permeable to staining reagent and cross-links these macromolecules so that they are stabilized and locked in position. Specimens for light and electron microscopy are commonly fixed with a solution containing chemicals that cross-link most proteins and nucleic acids. Fixatives kill the cells while preserve their structural appearance. The most widely employed fixatives are aldehydes such as formaldehyde and glutaraldehyde. Formaldehyde. Now discuss about formaldehyde. Reaction of aldehyde group with primary amines in proteins. Aldehyde group from this fixative reacts with primary amines in proteins present in sample, a specimen. Then glutaraldehyde, another fixative. It is also primary fixative, contains two aldehyde groups separated by 3 methylene bridges. These two aldehyde groups and the flexible methylene bridge greatly increases the cross-linking potential of glutaraldehyde over formaldehyde. Okay, it penetrates rapidly and stabilizes proteins by forming cross-links but does not fix lipids. Osmium tetroxide is used as a secondary fixative, reacting with lipids and acting as a stain. It is especially useful for fixing cell membranes since it reacts with the carbon double bonds present in many fatty acids. Following each fixation step, excess fixative must be washed out of the tissues. So this is all about fixative. Okay, if it carry 2 marks, squeeze it, make it coincide, and if in detail, the all information in front of you. So stay connected for next session.